In this video, I'm going to break down the infamous Randle cycle, aka the glucose fatty acid cycle, for you in depth and show you exactly how it is involved in metabolic and chronic diseases.
A report out today says 1 out of 10 people on Earth is obese.
Physicians are sounding the alarm after conducting a study that predicts the prevalence of diabetes in American youth.
We're talking about a disease called MASH that affects 1 in 20 adults in America, and this is a severe form of fatty liver.
While I know the mechanisms of the Randle cycle are a bit in depth overall, they are actually quite important to understand from my perspective because they are directly involved in the pathology of metabolic diseases. By understanding the mechanisms of the Randle cycle in the appropriate context, I think we can start to build targeted energy-based strategies to overcome metabolic disease. I do want to give a warning though, this is going to be pretty in depth as far as the cellular biochemistry. So just bear with me. I have a bunch of graphics to show you to try to make it make sense. And Ethan and I have gone through this structure so many times, trying to like whittle it down, break it down to try to get to the key points in understanding this and have it in a nice logical flow. So bear with me. I think it'll be really helpful because after I described it to Ethan, he said that he was able to really understand the Randle cycle quite a bit better. And so let's see how it goes. So there are two sides of the Randle cycle. We have the fatty acid inhibition of glucose oxidation, and then we have the glucose inhibition of fatty acid oxidation. So we're going to start with the fatty acid inhibition of glucose oxidation, and then we'll go to the glucose inhibition of fatty acid oxidation. When carbs and fats are oxidized, their electrons are removed and transferred to little batteries, essentially, called NADH and FADH2. This occurs in glycolysis and beta oxidation, which I don't have pictured, but we can see it happening here in the Krebs cycle. So we see here, both carbs and fats come in and they both get converted to citrate. And then as citrate moves through this process, the NAD plus here gets converted to NADH, and there's also FAD is in here, and it gets converted to FADH2. So these basically are storing the electrons that you're pulling off of the carbs and the fats. And again, both of them come in here at citrate. So once they start going through the cycle and you get some NADH and FADH2 from beta oxidation and glycolysis as well, you're basically storing electrons in these little batteries. Now, these little batteries are then brought into the electron transport chain, which is the second part of the mitochondria you need to know. So you have the Krebs cycle, the electron transport chain, and they power little pumps in the membrane that move H plus ions. And we can see this happening here. So you have these little pumps are called complexes, complex 1, complex 2, complex 3, you have cytochrome C and coenzyme Q, complex 4, and then complex 5 is ATP synthase. So these, the NADH you can see here and FADH2 drop off these electrons to the pumps, and the electrons basically move across these kind of like a wire, and they drive an electrical reaction. They power these pumps to move the protons across the membrane. So what happens is, as these protons, these H plus ions, that's what the protons are, as they move across the membrane, you're basically saturating this intermembrane space. So you have the outer mitochondrial membrane and the inner mitochondrial membrane. You're saturating this with the protons, and you're basically building up a gradient. So you're basically, think of this like a water pump. You are pumping tons of water into here. And then what eventually happens is the water, as it builds up into here, there's a little dam here, ATP synthase. And ATP synthase, you can think of like a big water wheel here. When the protons move down ATP synthase, that water wheel generates energy in the form of ATP. So you're basically creating your own little hydroelectric dam inside your cells to power your energy production. Now, something that's really important, FADH2 drops off its electrons at complex 2. NADH drops its electrons at complex 1. Both complex 1 and complex 2 use coenzyme Q to then transfer the electrons to complex 3. So they're basically like two siblings that don't get along with each other, and they're fighting over coenzyme Q. They're fighting over this Game Boy or whatever it is here, and that it's to drop off their electrons. Now, this is really important to understand, because this is where the difference with carbs and fats starts to become pretty significant. The difference in carbs and fats is centered around the NADH and FADH2 production. So fatty acid oxidation creates basically a ratio of 2-to-1 NADH to FADH2, whereas carb oxidation creates a ratio of 5-to-1 NADH to FADH2. Now, the reason that we get these different ratios of NADH and FADH2 between carb oxidation and fat oxidation is because during carb oxidation, if we look here at this graphic, we have glycolysis. We see that one molecule of glucose, as it goes through glycolysis and gets converted into pyruvate, where the pyruvate eventually goes into the Krebs cycle, produces two molecules of NADH. So we get two NADH for every molecule of glucose as that glucose runs through glycolysis. However, when we look at fat oxidation, where it's called beta oxidation, when a free fatty acid undergoes beta oxidation, you get one FADH2 and you get one NADH. So in the Krebs cycle, we're always going to get one FADH2 and three NADH. So if you take the two NADHs that you get from glucose and you add that to the three NADHs that you get from the Krebs cycle, you're going to have five NADH total and only one FADH2. Whereas when you look at the beta oxidation, you get the one FADH and the one NADH from the beta oxidation. When you add that to the Krebs cycle production of NADH and FADH2, you see that you have two FADH total and four NADH total, which gives you that 2-to-1 ratio rather than that 5-to-1 ratio that you see with carbohydrates. So it's how the carbs and fats are processed actually changes that ratio. And I want to give you guys a look here so you can see what exactly is going on and the differences between glycolysis and between beta oxidation. Now, as I mentioned, NADH goes through complex 1, while FADH2 goes through complex 2. When you start to have a lot more electrons flow to FADH2, which you see in fats, because you have only a 2-to-1 ratio of NADH to FADH2 versus carbs, where you have a 5-to-1 ratio of NADH to FADH2. Complex 2 starts to hog CoQ10 and steal it from complex 1. And so its electrons go, complex 2 drops electrons off to coenzyme Q, which goes to complex 3. And then complex 1 is sitting there and it has the NADH and it's trying to drop off the electrons, but it can't because it's already loaded with electrons. And it basically gets jammed up because it can't bring it to coenzyme Q. So it creates this circumstance where complex 1 is kind of pissed off. Complex 1 is like, well, I didn't get my turn, basically. And then what happens is the NADH keeps flooding in, and then the NADH cannot drop off more electrons at complex 1 because complex 1 is already full because it couldn't give it to coenzyme Q. Now, this basically leads to a circumstance where complex 1 starts to produce a lot of reactive oxygen species because it can't drop its electrons off. So it's getting pretty angry. It's getting pretty pissed off. And you start getting this ROS without that flowing of electron appropriately. So fatty acids drive this. On top of that, the NADH that was supposed to get there starts to build up. And so when you have a buildup of NADH, since it's created from NAD+, you don't actually get NAD+ anymore. And now this is a huge problem. Now, why is this a huge problem? So the enzymes in the citric acid cycle, like isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, and pyruvate dehydrogenase, not directly in the citric acid cycle, but it connects to the citric acid cycle, and it's in the mitochondria. Pyruvate dehydrogenase, all these enzymes require NAD+, but it doesn't have NAD+. Why doesn't it have NAD+? Because complex 1 is jammed up and it can't drop off its electrons, and so the NADH, which is NAD+ plus the protons, the H plus ions, is just sitting here. And it's like, well, we can't get converted to NAD+ because we can't drop our electrons off at coenzyme Q, and we can't give the electrons to complex 1. So what do? So you have a circumstance where now these enzymes, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, and most importantly, pyruvate dehydrogenase are kind of screwed. They can't undergo their reactions because they don't have the products they need for those reactions to occur. And the other thing that happens is you get a backup here on this right side of the Krebs cycle where you get a backup of these intermediates because the enzymes aren't working effectively, and you get a buildup of citrate, you get a buildup of acetyl-CoA. And then essentially this buildup of acetyl-CoA, the buildup of the NADH, these components directly shut down pyruvate dehydrogenase, which we'll see in a second is very important because pyruvate dehydrogenase brings carbohydrates, or pyruvate, which is a product of carbohydrates, into the mitochondria to enter into the Krebs cycle. So the other thing that happens, and it's important to note, and it's a little bit of a side piece, but citrate, acetyl-CoA, and NADH, these components here, so the citrate, this acetyl-CoA, and then we have the NADH buildup, they activate a protein called pyruvate dehydrogenase kinase. And pyruvate dehydrogenase kinase, we can see here in this image, we see PDH kinase. It makes pyruvate dehydrogenase inactive. It shuts down pyruvate dehydrogenase further. So pyruvate dehydrogenase is already shut down because it doesn't have enough NAD+. It's already shut down by the acetyl-CoA, the citrate, and then the high amounts of NADH. And then PDK is activated, and it further shuts it down. So basically, it shuts the door to carbohydrates going into the Krebs cycle inside the mitochondria because they can't flow through pyruvate dehydrogenase effectively. And then basically, they get stuck going either to glycogen storage, they get stuck to going to creating fatty acids in the cell, or they get stuck into getting converted to lactate or moving in the pentose phosphate pathway or the hexosamine biosynthetic pathway. So they get kind of shunted everywhere else. So with this, PDH is shut down, and we have this some of this buildup of citrate, as well as acetyl-CoA and some of these components. Now, what winds up happening is that citrate, we can see in this graphic, then leaves the mitochondria. It goes out and from the mitochondria into the cytosol of the cell, which is basically the interior portion of the cell, but not in the mitochondria. The mitochondria is kind of like its own little island in the cell. And the citrate then goes to inhibit these enzymes in glycolysis. So glycolysis is a process that takes glucose, and once it's brought into the cell through GLUT4, or it could be brought into the cell, like if the cell doesn't need GLUT4 in that circumstance, because it's an insulin independent cell, then basically the glucose runs through glycolysis through hexokinase and phosphofructose kinase, which are two of the biggest enzymes, two of the major enzymes. There's other ones. And the citrate inhibits hexokinase by about 20 to 30%. And it also inhibits phosphofructose kinase by 40 to 60%, where it completely shuts down, or PDH is already completely shut down. So the whole processing glucose into the mitochondria, into the Krebs cycle, is shut off by the fatty acid oxidation because of this change in the NADH to FADH ratio, as well as the effects at complex 1, and then the buildup of acetyl-CoA, the lack of NAD+, because you have so much NADH. So then basically you're not able to use the carbohydrates as well now. Now there's something important to note here, and it gets a little bit, it's a little bit specific, but it will become more important when we start to talk about carb inhibition of fat oxidation. And this is why they don't just like inhibit themselves to some extent, as some other people have tried to mention when they talk about this cycle. But essentially, when citrate goes back into the cytosol, so that citrate can eventually be converted into malonyl-CoA. The problem with this is that malonyl-CoA, while you're burning fatty acids, would inhibit carnitine palmitoyl transferase 1, which is the major transporter that brings fats into the mitochondria for oxidation. So the question is, how come when you're burning fats, that citrate doesn't get converted to malonyl-CoA, so the fats aren't inhibiting their own oxidation? And the reason why is the enzyme that converts citrate into acetyl-CoA is an enzyme called ATP citrate lyase, which we can see here. Now, ATP citrate lyase needs CoA in order to take the citrate and the CoA and some ATP and produce acetyl-CoA, where the acetyl-CoA then gets turned into malonyl-CoA via an enzyme called acetyl-CoA carboxylase. If you don't have enough CoA for ATP citrate lyase to do its job and convert citrate to acetyl-CoA, then you can't get malonyl-CoA. And so what's stopping the ATP citrate lyase? What's pulling all of the CoA out of the cytosol so that the ATP citrate lyase doesn't have the CoA it needs? Well, when you burn fats, or when you are oxidizing fats, the fats have a CoA group added to them to make a fatty acyl CoA, and then they are brought in as a fatty acyl CoA into the mitochondria through carnitine palmitoyl transferase 1. This CoA is added to the fats in the cytosol of the cell. So essentially, the fatty acid oxidation is depleting the cytosol of the CoA. So ATP citrate lyase is unable to take the citrate and convert it to acetyl-CoA by adding the CoA group, because there aren't the CoA groups available. So this leads to that buildup of citrate, which we're going to see doesn't happen when carb oxidation inhibits fat oxidation for this reason. And then that citrate is allowed to oxidate or to inhibit glycolysis. This is really important to understand Because if you don't understand this mechanism, then you start saying stuff like, well, when fats leak citrate into the cytosol, they block hexokinase and phosphofructokinase. Or when carbs leak citrate into the cytosol, it blocks hexokinase, phosphofructose kinase as well, because you don't understand that there's this flow towards malonyl CoA and that you have a regulating enzyme that gets adjusted based on carb oxidation and fat oxidation that shifts the citrate to malonyl CoA when you have carbs, versus when you have fats, it stays at citrate because you have the depletion of the CoA pool. And so that's where you get these weird interpretations of the Randle cycle that we've seen from some of the low carb community. Now, all of this together basically results in the inhibition of glucose oxidation by fatty acid oxidation, with the shunting of that glucose towards glycogen stored, or it can be converted to fatty acids, or it can be converted to lactate through lactic acid fermentation, or or it can be pushed through the pentose phosphate pathway or other pathways, but the carbs basically aren't making it into the mitochondria to be converted into fuel sources. Now, this brings us to the glucose inhibition of fatty acid oxidation. And so there's some context here that also becomes important, and that's around insulin and lack of carbohydrates in the diet. So under circumstances when you don't have insulin present, your body automatically oxidizes fatty acids, starts to increase the release of fatty acids as a primary fuel source, and it saves the carbohydrates for your central nervous system, which needs the carbohydrates, and other tissues can get by on the fatty acids. As I mentioned, it's still not ideal to run primarily on fatty acids because of the mechanisms that I described at the beginning of this video. But when you eat carbohydrates, it stimulates the release of insulin. And insulin is that signal to the body to be like, hey guys, we got the carbs, party's here. So what insulin does is it starts to increase the recruitment, we can see in this image here, of the GLUT4 transporters to the cell membrane of the insulin-dependent tissues like the muscle tissue, the fat tissue, and the liver. And then basically the fat tissue, the muscle tissue, and the liver can start to uptake the circulating glucose that's been brought in. And what this does is this will lower the release of free fatty acids from the fat tissue. And so the circulating fuel source will be predominated by glucose, and the other cells will not get the same influx of fatty acids. They'll get the glucose, even the insulin independent cells and tissues. The liver stops producing glucose through gluconeogenesis because it doesn't have to do it anymore because it has glucose coming in. And then the muscle tissue and the fat tissue start to take up that glucose as well as the liver. The liver and muscle will make some glycogen, and then the fat tissue will start to store some as fat so that when you aren't eating, you have some stores ready to go so you can last between those times that you don't have the energy supply in. So those are your storage sites, is your muscle tissue, your fat tissue, and your liver. Those are major storage organs. And this is why they have the GLUT4 transporter there, because the GLUT4 transporter and insulin signaling is like, hey guys, we have a party, we have influx, we just got paid right now. And then that's like, these are going to be the deposits into the bank. Those are our savings so that we don't have to live hand to mouth next time we don't have a meal and have to take out a loan by using the stress hormones. So your body has this system built in place. So it has some deposits in its bank accounts, your fat tissue, your liver, and your muscle tissue. Now, the other thing that insulin does, besides just increasing GLUT4 and allowing the cells to uptake glucose, is insulin also changes the activity of certain enzymes in the cell that help to inhibit fat oxidation. So we have a graphic here. This is the similar graphic that I was showing you before. So we have beta oxidation, and you also have glucose oxidation. So fat oxidation, glucose oxidation going on here in the mitochondria. And then here we have the cytosol. Now, as we mentioned in fat oxidation, same thing happens in glucose oxidation. You have citrate will leak out of the mitochondria, and the citrate leaking out of the mitochondria can either be converted into acetyl-CoA, or it can stay as citrate and inhibit the carbohydrate, the glycolytic enzymes. So what winds up happening when insulin is present, insulin activates the enzyme acetyl-CoA carboxylase to convert the acetyl-CoA in the cell to malonyl-CoA, which shuts down carnitine palmitoyltransferase 1, and it stops the fatty acid flow into the mitochondria so that you can allow the carbohydrate flow to come in. On top of that, there's another enzyme that's not pictured here. It's called malonyl-CoA decarboxylase. So that enzyme actually breaks down malonyl-CoA, right? Acetyl-CoA carboxylase carboxylates or adds a CO2 group to, or the carboxy group to the malonyl-CoA. And then the malonyl-CoA decarboxylase just removes that group. So it removes that group and gets rid of the malonyl-CoA. So insulin increases malonyl-CoA by increasing production via acetyl-CoA carboxylase and decreasing breakdown by inhibiting malonyl-CoA decarboxylase, which increases the amount of malonyl-CoA in the cell and directly blocks fatty acid oxidation. So insulin forces carb oxidation by inhibiting fatty acid oxidation. This is pretty important because then that allows the carbs to come in, because it's like, well, how is the carbs switching, right? If you have a bunch of fats floating around all the time, when the electron transport chain gets stopped because you have too much FADH over reducing coenzyme Q from complex 2, how do you then switch it over to the carbs? And basically, the insulin is lowering the release of free fatty acids from the fat tissue. And then on top of that, the insulin is forcing the inhibition of fatty acid oxidation mitochondria by blocking carnitine palmitoyltransferase 1 while increasing the uptake of glucose. So insulin's a potent stimulus to force carbohydrate oxidation and minimize the release of free fatty acids. Now, when the fats aren't being oxidized, the next question is like, well, what happens to them? Where do they go? Basically, you can see here, the fats get converted to triglycerides. So the fats start to become esterified, they get converted to triglycerides, and they basically aren't brought into the mitochondria anymore. They basically go towards storage. Now, when the fatty acids are out of the way with the insulin, the glucose is then able to move through glycolysis, come down here, and it creates pyruvate. That pyruvate then directly stimulates pyruvate dehydrogenase. So pyruvate itself stimulates pyruvate dehydrogenase, which is the main enzyme that brings the pyruvate, which is the product of carb metabolism, into the Krebs cycle, and then it runs through the Krebs cycle to produce the NADH and FADH2, so that you can bring that into the electron transport chain and produce ATP. Now, the other thing that happens is when you inhibit the fatty acid oxidation and you basically lead to a circumstance where you don't have all the FADH2 going to complex 2 and over reducing coenzyme Q, you start to get more NAD levels, right? Because NADH can now be utilized at complex 1, and you can recycle and get your NAD+. When you basically have the insulin present, you stop the fat oxidation, you have less FADH2, it stops hogging coenzyme Q, and then complex 1 can start to work. And then now you start to get NAD+. That's important because that NAD+ then allows isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and pyruvate dehydrogenase to further work, which allows things to flow through here. You get more CoA, so pyruvate dehydrogenase can start to work again or start to function again. And then essentially you also will start to inhibit with some of these components the pyruvate dehydrogenase kinase. And then once the pyruvate dehydrogenase takes the pyruvate, converts it to acetyl-CoA, that acetyl-CoA will mix with oxaloacetate and run through the Krebs cycle, and again, produce the NADH and FADH2. But you will now have a 5-to-1 ratio of NADH to FADH2, which won't create that blockage at complex 1 from the electrons running primarily through complex 2 and hogging coenzyme Q, because now you have much more NADH with that carb oxidation. Now, this brings us back to the point that I was talking about with the ATP citrate lyase and how carbs inhibit fat oxidation through malonyl-CoA. Now, with carb oxidation, you will still get some citrate that'll flow from the mitochondria back into the cytosol of the cell. Now, since fats aren't being oxidized right now, then they are being re-esterified. They're converted to triglycerides and they're stored. The cytosol now has CoA. So now ATP citrate lyase, which we see here, that requires CoA, can take that citrate and convert it into acetyl-CoA. And when it's converted to the acetyl-CoA, we have insulin stimulating acetyl-CoA carboxylase, and you also have the acetyl-CoA, which can now convert to malonyl-CoA, and that malonyl-CoA further inhibits carnitine palmitoyltransferase 1. So you have a circumstance where now you have the carb oxidation by itself, as you have the carbohydrates come in, is inhibiting fatty acid oxidation alone. The insulin signaling is also helping that. So you have this shutdown, and this is why we don't see, well, carbs release citrate back into the cytosol, but why doesn't the citrate here inhibit hexokinase or phosphofructose kinase? Well, it doesn't inhibit it because it gets converted downstream to malonyl-CoA because there's enough CoA at ATP citrate lyase to actually allow for the cell to convert the citrate into acetyl-CoA, because it has the products it needs to do that reaction. Now, all of this together creates a circumstance where the fatty acid oxidation is inhibited, the fatty acids are shunted towards triglyceride and stored, and the glucose is allowed to come into the cell, become the primary fuel source, and be oxidized and converted into ATP. So now we have an understanding of how carbs inhibit fat oxidation and how fat inhibits carb oxidation. The question is, how does all of this relate to diabetes, insulin resistance, obesity, fatty liver, and overall metabolic dysfunction. Diabetes, insulin resistance, obesity, fatty liver, cardiovascular disease, etc., are all characterized by high levels of fatty acids in the blood, as well as deposits of fatty acids in tissues like the liver, the muscle, and the fat stores. Now, these fatty stores accrue over time due to poor diet, micronutrient deficiencies, hormonal imbalances, microbiome disruption, toxic exposure, inactivity, etc. And a quick side note here is that correcting these things are, in my opinion, the best way to actually correct these states with diet, supplementation, appropriate physical activity, lifestyle adjustments, etc. But either way, these fatty acids that are built up in the tissues and also constantly being released from these tissues, particularly the fat tissues, can lead to metabolic issues and difficulty in utilizing glucose and creating that insulin sensitivity due in part because of this Randle effect, because you have the cells are not responding to insulin and they are oxidizing fatty acids. They're stuck in this fatty acid oxidation, which further inhibits that glucose utilization. Now, even worse overall is the buildup of the fats in these tissues and the constant fatty acid oxidation in these tissues. And with ROS production, the general metabolic dysfunction that gets created actually directly leads to impairment in that insulin signaling. Now, this leads to the inability of the muscle tissue to actually clear the glucose from the bloodstream, even when insulin is present, leaving more glucose in the bloodstream. It leads to the inability of the fat cells to take up glucose, as well as the fat cells are unable to stop releasing free fatty acids in the bloodstream because they're not responding to the insulin. And now you have both more glucose and more fatty acid in the bloodstream. And it also leads to the inability of the liver to stop its production of glucose via gluconeogenesis, even with insulin present, leaving more glucose inside the bloodstream. So you have a circumstance where you have high glucose, high fatty acids, and then the high insulin in the bloodstream. The cells and tissues are not responding to that insulin, and they're being overloaded. They're already kind of overloaded with fatty acids for a variety of different reasons, as I mentioned, but then they're also being bombarded with both substrates simultaneously. And as we saw, that doesn't work well because each cell can only oxidize one substrate at a time. And if they're already oxidizing fatty acids and they're getting presented with glucose, they're going to have difficulty with using that glucose. And also, they're not going to be able to respond to insulin effectively because they can't. The insulin is a signaling hormone. It tells the cells to do X, Y, and Z thing. But if the cells don't have the resources or have the ability to respond to insulin, then they're not going to do it regardless of if the signal is present. And that's what you see with insulin resistance. Now, ultimately, this obviously leads to the circulation of both high amounts of fatty acids and glucose in the bloodstream. And this creates a circumstance where we know that both glucose and fats can't be metabolized at the same time. And so this creates a circumstance where you have high amounts of oxidative stress because the cells are stuck oxidizing the fatty acids and they're also being presented with glucose. And they're like, well, what do we do? What do we do here? They're kind of being overwhelmed by this metabolic dysfunction. So this ultimately, the oxidative stress inside the mitochondria and inside the cell, because they're overloaded with the substrate, directly damages the mitochondria and directly damages the cellular components, the interior components of the cell, the endoplasmic reticulum, the ribosomes, the protein structures, etc. Now, long term, this can lead to a circumstance where you get progressive mitochondrial dysfunction and progressive cellular dysfunction because that oxidative stress is directly damaging their structure. So if you have your mitochondria, think of it like an engine. If the engine becomes destroyed, if the belt on the engine's cut, if the spark plugs are taken out, if the spark plugs don't work, the engine's not going to work. And the same thing happens in the mitochondria. If the mitochondrial membrane starts to get destroyed or its proteins start to get destroyed, and then if it's DNA, the mitochondrial specific DNA starts to get damaged in and of itself, then you wind up running into a circumstance where the mitochondria can't function appropriately, and then it has difficulty in actually being able to recover itself or produce new viable mitochondria. Now, as the cells starts to get damaged, as the mitochondria starts to get damaged, you continue to get this backup more and more of the fatty acids and the glucose in the bloodstream. And you also have the oxidative stress coming from these cells, and the body has a generally high oxidative stress burden. You get a backlog of the substrate into the bloodstream. Now, this creates quite a bit of problem. Why is that? Well, first things first, with the high blood glucose levels, the pancreas is like, well, what do I do? Let's put out insulin. We got to get the blood glucose levels down. The pancreas is responding to this, and it keeps trying to put out the insulin to try to bring those blood glucose levels down. But over time, the pancreas becomes fatigued. The pancreas starts to develop oxidative stress from trying to keep up with the insulin production that's supposed to do under these circumstances. And then eventually, over time, over long-term type 2 diabetes, the pancreas begins to fail because from accumulating excessive amounts of damage. Now, on top of this, the vasculature, it's exposed to both the oxidative stress from the cellular components that are being damaged from the impaired mitochondrial function and the delivery of both fuels and the overload of those fuels. And then on top of that, it's now the bloodstream is backlogged with free fatty acids and glucose, and the endothelial cells of the vasculature are getting beat up by the high amount of both substrate on top of the oxidative stress, which then damages the cardiovascular system over the long term. Then further, you have organs that are dependent on the cardiovascular system, like your kidneys, like your eyes, like your central nervous system, your peripheral nerves. And you see in people with type 2 diabetes, they get kidney disease, they get blindness, neuropathy, nephropathy, and they also get peripheral neuropathy, which is damage to the peripheral nerves. And it's related to the metabolic dysfunction as well as the high amount of substrate, damaging the supply of nutrients and damaging the vasculature, the microvasculature to these areas, so that now they are unable to actually get enough of the substrate and oxygen to them effectively because of that progressive damage. And you eventually get kidney damage, blindness, you get peripheral neuropathy, etc. And also, you also see diabetics will get when I was working in the ICU, I saw this all the time, these horrific wounds that won't heal because those tissues don't have the circulation supply and the appropriate metabolic function to actually heal the correct way. So they just stay and they start to get infected, and the immune system doesn't work as well, or it doesn't work well either. Again, through this impairment or the central impairment of mitochondrial function. So ultimately, the Randle cycle is a problem, not necessarily from your individual meal per se. The Randle cycle is a problem when you have metabolic dysfunction, when the cells are overloaded with substrate and they're not processing the substrate effectively, and they're not responding to the insulin signaling anymore. And basically, you have this backlog in the cell of both fats and carbs. And in the bloodstream, you get the backlog of both fats and carbs. And this is why, whether you do low carb, whether you do high carb, whether you just go on a general caloric deficit and you clean up your diet, a lot of metabolic markers and elements improve, because what you're doing when you're losing that body fat and when you're in that deficit state is you are pulling out that backlog of substrate. You're clearing out the backlog. Your engine is flooded with fuel, and you're clearing the engine out so that it's not flooded anymore, so that you can start to drip that fuel back into the engine and get it firing again appropriately. And also in the meantime, and this is something from pro-metabolic bioenergetic perspective, what I'm looking to do is I'm looking to make sure that we manage that oxidative stress. We minimize that damage to the mitochondria. We want to make sure the mitochondria have all of the vitamins and minerals they need so that those proteins and the enzymes in the Krebs cycle at the electron transport chain can function appropriately. We want to make sure our mitochondrial membrane is strong. It's not loaded in high amounts of polyunsaturated fats that lead to an increased risk of damage from oxidative stress. We want to make sure our vasculature is strong. We want to make sure we don't have toxins coming in, poisoning the mitochondrial function, or poisoning or creating an inflammatory response that then shifts hormones or directly impairs metabolic function as well. So we want to fix all of these things so that we can clear that backlog of substrate, take the pressure off the mitochondria and the cells, and allow things to work appropriately, where you take in the fuel and it's oxidized appropriately to energy, and you're not getting this buildup, and you're not forced into the circumstance where you're presented with both fats and carbs simultaneously, and the cell doesn't know what to do with both those fuel sources, and it starts creating massive amounts of dysfunction and massive amounts of cellular damage over the long term. So with this, I showed you the Randle cycle mechanisms in depth. I tried to put them into context, but if you want to know what to do with your diet, I'd recommend you check out this other video I did on the Randle cycle over here.